Okay, this is GATE 2022 Life Sciences, Section S Microbiology. Question number 67. Which one of the following mutagens convert DNA's adenine to hypoxanthine? A. Ultraviolet light. B. Mitomycin C. C. Methylmethane sulfonate. And D. Nitrous acid. Okay, so there are various mutagens, okay, which are able to mutate the DNA inside the cell. Okay, we'll briefly go over each types of different mutagens. So mutagens are broadly classified into three types: physical mutagens, okay, physical mutagens, chemical mutagens, and chemical mutagens, and biological mutagens. Okay, so in physical mutagens we have different ionizing radiations, which includes gamma rays, UV rays, alpha particle, beta particle, etc., and non-ionizing radiations. Okay, the difference between ionizing and non-ionizing radiations is pretty simple. They are able to ionize the atoms present in the DNA. Okay, so these are the different examples: gamma rays, X-rays, UV rays. uh, you all must be familiar with the UV rays, use of UV rays as a source of physical mutagens. Okay, and in chemical mutagens there are various chemicals, like these could be either alkylating agents. So in some way they'll alkylate the purines and pyrimidines in the DNA. Deaminating agents, they can either deaminate the purines and pyrimidines residues. They can eliminate the amino groups on the purines and pyrimidines. And then we have intercalating agents. Okay, we are also familiar with the intercalating agents. So we are all familiar with ETBR. ETBR is the most common example of a intercalating agent. Okay, so there are, apart from this, the chemical mutagens also contains various alkylating and deaminating agents. Okay, so these are the examples. So mustard gas and nitrous acid gas, these all are deaminating agents. Okay, whereas ethyl methyl sulfate is example of alkylating agent. So they add the alkyl groups. Okay, they add this ethyl or methyl group to the purines and pyrimidines in the DNA. Then we have biological mutagens. So biological mutagens are also very common. These could be either virus, bacteria, or transposons. So virus are able to intercalate the viral genome into the normal DNA of cell, and which can also lead to mutation. Bacteria has various plasmids, right, and these plasmids are also able to intercalate into the genomic DNA to cause mutations. And then we have transposons. Transposons are nothing but jumping genes. So these set of genes are intercalated into the human genome. Okay, so in the question, in the options we have ultraviolet, mitomycin C, methane sulfonate, and nitrous acid. So from the question we know that this is an example of a physical mutagen, right? Physical mutagen, and these three are examples of chemical mutagens, right? So chemical mutagens are those mutagens which convert the DNAs, which convert the purines or pyrimidines in the DNA. Okay, and when these purines and pyrimidines are converted, they are either alkylated or they are either deaminated. So what will happen if they are alkylated or deaminated? During replication, if any purine or if suppose the is um, adenine is deaminated and converted into hypoxanthine. Okay, so adenine will no longer be read in the during replication process when these two strands are separated. Okay, and then these two strands are again replicated, a new replication. In place of adenine, now we have a hypoxanthine because of deamination. Okay, because of deamination we have a hypoxanthine, and due to this hypoxanthine here, in place of hypoxanthine there will be another different nucleotide. So it should be T here. T should be added in opposite of A. Now in place of T there could be any other nucleotide could be added. Okay, so this will lead to a point mutations in DNA. Okay, so we'll one by one look into all these different mutagens. So first we have ultraviolet light. So what happens? Ultraviolet light are famously known to create pyrimidine dimers. Okay, so UV light are mostly known to create pyrimidine dimers. So we are familiar with the term of thymine dimers, right? Thymine dimer. So what happens when ultraviolet light is reflected on the DNA? The T's, adjacent T's, they form a dimer with each other. Okay, like this, they form a dimer, and what will happen? There will be a abnormal DNA, and this abnormal DNA will not be stable, and it will lead to breaks in the DNA, and these breaks can be repaired. Or it could be mutated. Okay, so it can be repaired by error prone or error free. If it is error free, we don't have any problem, error free repair. But if it is error prone, we'll have the mutated DNA. Okay, our DNA will be mutated if this break is not properly repaired. If it is error prone, we'll have a mutation here. Okay, so UV light is mainly responsible for formation of pyrimidine dimers, and most probably it is always thymine dimers. Okay, then we have chemical mutagen. The another example was mitomycin C, right? We had option B was mitomycin C. So this is the structure of mitomycin C. What happens? Mitomycin C forms an adduct with the DNA. And what is this adduct? So this adduct term is very important. Okay, it is very familiar. Many of the chemists use this term adduct. So what happens? So we have, yeah. So adenine has a amino group. Okay, adenine has a amino group here, whereas guanine has a carbonyl group here. Okay, so this is a guanine. Okay, this is G adduct. This is the guanine adduct. So what happens when we add mitomycin C? Mitomycin is a chemical. When we mix it with DNA, right? When we mix it with DNA, what happens? It forms a adduct. So this is our mitomycin C. Okay, it forms a adduct, guanine adduct. And by adduct I mean it forms a complex with guanines in the DNA. Okay, so and this can lead to a DNA damage. So guanine is a small molecule, and when there is formation of this big adduct, it creates kind of a kink. Okay, it creates a kink in the DNA. Okay, it creates a kink like this in the DNA. This is where the adduct is formed. Okay, this is the G adduct. It creates a kink kink. And what happens when there is a kink? There when the polymerase recognizes the kink here, when the replicative polymerase is replicating the DNA, okay, it is replicating the DNA from here. When it comes across this kink or the adduct, it will just halt. Okay, it will halt and it cannot bypass this adduct. Okay, this is very important. The replicative DNA polymerase is not able to bypass this adduct. So then this polymerase will fall off. Okay, this polymerase will fall off, and then we'll have these are the different repair polymerases. Okay, repair polymerases, this is very important. These are the human repair polymerases: Pol eta, kappa, okay, rev1, pol. Okay, these are the different repair polymerases. So these are recruited because these so there is a difference in the structure of a replicative and repair polymerase. They have a much wider clamp like structure. Okay, replicative clamp is much larger, which is able to accommodate this adduct. So these TLS DNA polymerases can bypass this. Okay, they are able to bypass this adduct, right? They are able to bypass this adduct, and then after bypassing this repair polymerases, they'll fall off. They'll fall off, and then the regular DNA replication will be again continued by the replicative DNA polymerase. Okay, so this process is known as translesion DNA replication. Okay, so there is a lesion here. Okay, there is a lesion here, and these replicating enzymes are known as repair polymerases or TLS DNA polymerases. Okay, they are able to bypass this adduct, and while bypassing, okay, they don't have much proofreading domain on them. The reading domain is very weak. Okay, so when they are bypassing, they'll add random nucleotides. Okay, so they'll add some random nucleotides here. There will be addition of, first of all, there will be addition of extra nucleotide, and the nucleotide addition will also be random. Okay, and both of these will lead to mutation, right? If there is one extra nucleotide, it will lead to the change in the reading frame. Remember the reading frame, what I was talking about? It will change the reading frame of the whole DNA, and it will also have random nucleotides added against the lesion. Okay, and so this process is also very important. Why? So if the replicative DNA polymerases halts, if there is a halt here and the replication is not continued, the cell will ultimately die. Okay, the cell will die. Thus this translesion DNA synthesis is very important. Okay, so in spite of it causes the formation of various mutations, but it is important that the replication is not stalled. If the replication is stalled, the cell will die. Okay, so it is very important that the replication is not stalled and the lesion is bypassed. Okay, lesion is bypassed. So this is very important process in the cells. Okay, translesion DNA synthesis is very important process in the cell. Okay, and the again repair can be error free or error prone. Okay, so it is very important to bypass these lesions, and hence this process is very important. So mitomycin C, what it does, it forms a G adduct. Okay, it forms G adducts. This is very important. It forms G adducts in the DNA. So the next option was methyl methane sulfonate. Okay, So methyl methane sulfonate. So again we have a guanine here, right? And this is the methyl methane sulfonate. Okay, so we have a methyl, methane and a sulfonate group. Okay, this is the methyl methane sulfonate. And what it is doing, so when methyl methane sulfonate is mixed with the DNA, this is our DNA. Okay, this is the DNA. What is happening here, the guanine, as you can see, we are getting methylated N7 methylated guanine. Okay, there is methylation at the N7 position. So we have a methylation of guanine. Electrophilic attack on the methyl methane sulfonate, and we have a methylated guanine. Okay, so this is also an example of a chemical mutagen. Okay, methylation of guanine is not normally seen, it is a regulated process. And inside the cell we have S-adenosyl methionine, right? We have SAM, S-adenosyl methionine. These molecules, they add the methyl group on the guanine, and this is not a natural process. This is a chemical mutagen which methylates guanine. This is very simple methylation of purines. And then we have the final one, nitrous acid. So we have a normal parent DNA. Okay, when we add nitrous acid, this is a nitrous acid formula, HNO2. What happens? Nitrous acid is induces adenine. It converts adenine to hypoxanthine, right? It converts adenine to hypoxanthine. So we have a adenine here. Now it is converted to hypoxanthine. Okay, till here we have a altered parent DNA. Till here we have a altered parent DNA. During the replication process, what will happen? Against hypoxanthine, now we have a C. Okay, so we have a C here. So actually it should be a strands are separated by helicase, right? This strand is unaltered, whereas this strand is altered. And against this strand we should actually have a T, right? But now we have a C. We have mutation of C, because against hypoxanthine pairs with cytosine. Okay, hypoxanthine pairs with cytosine, so we have a cytosine added. So the replicative enzyme adds cytosine against hypoxanthine, right? This is a transversion mutation, right? And then further mutated granddaughter DNA will have a GC. So from TA we have a GC mutation, right? From TA we have now a GC mutated DNA. And again to the hypoxanthine there will be further addition of the C. Then the mutation will be sustained in the cell. Okay, so what is happening? Nitrous acid, what it is doing, it is a deaminating agent. Okay, it converts adenine to hypoxanthine, and thus there is a mutation. Okay, TA to GC. So coming back to the question, UV forms a thymine dimer. Mitomycin C, what does mitomycin C do? Adduct formation, G adducts formation. And then methyl methane sulfonate, we have methylation of guanine. This is also guanine. And nitrous acid, it converts, remember this is very common, adenine to hypoxanthine. Okay, this is very common conversion. And we have nitrous acid also, and then we have bromouracil, 5-bromouracil. You guys should go and read about 5-bromouracil. So you guys go and read about this 5-bromouracil. Okay, it is very common mutation. it's an analog, and again it is a thymine analog, and a different nucleotide is added against it. Okay, go and read about 5-bromouracil. So nitrous acid and 5-bromouracil, this is also one of the most common mutagen. Okay, so the answer is D, nitrous acid.